Like I've never been in the
Not a chance in hell.
Good morning, class.
never been.
Or, I'm sorry, good afternoon, class. How's everybody doing today? Good. If you have a moment, please go ahead and sign in to the Turning Point session.
Going to the moon, Kim. We're going to the moon.
Okay, so the topic for today is kind of an extension of our discussion of amino acid metabolism. We're going to talk about the urea cycle today and some of the intertissue relationships between the liver and everything else in the body, essentially. The urea cycle takes place in the liver, but its job is to get rid of excess nitrogen from all over the body. Okay. The objectives are here, and I think that that's pretty self-explanatory. I have a case on kwashiorkor at the conclusion of the session. I'm not sure if we'll get that, but hopefully we'll at least define it and what it is. Hepatic encephalopathy, that's I don't think I have that anywhere explicitly in your slides, but I'm gonna, when it comes up, I will define it, and hopefully you'll get that down. Okay. So as far as the outline for today, first we'll talk about the turnover of amino acids in tissues and how excess nitrogen gets from peripheral tissue like skeletal muscle to the liver for the urea cycle. Then we'll talk about the enzymes of the urea cycle and what its function is, and some information about how it's regulated. And then we'll conclude with a discussion and some cases on disorders of the urea cycle. Okay. All right, and those cases, this is the first one. Any thoughts? Could be anything, right?
Pretty much.
Could be just a fussy baby, right? Okay, this one is a little bit more, I guess, telling. So we've got hyperammonemia, okay, elevated ammonia in the blood, and that's a classical hallmark of all of the urea cycle disorders. Okay. Then this one, this is not a urea cycle disorder case, but it's a case of a disorder of nitrogen metabolism and an amino acid metabolism. Did I include this in your downloads?
At the very end.
It is at the end. Okay, good. I kind of shifted around what I was going to do in between when I put your thing uploaded, and now. Okay, should we do a few clicker slide? Excuse me, a few clicker questions to get started. And this will kind of get to the go back and revisit some old stuff and then some more recent stuff. Okay. All right, very good. So glucagon is the G protein coupled receptor. If this question was asking about the insulin receptor, that would be the tyrosine kinase receptor. Okay. Okay, further on to the G protein coupled receptors. Okay, everybody got a best answer? Okay, a plurality of respondents answered correctly. Answer A. So the activation of the heterotrimeric G proteins, that occurs when the alpha subunit drops off a GDP and exchanges it for a GTP. Okay, so the GTP bound alpha subunit is then active, and it becomes inactivated when the alpha subunit hydrolyzes the GTP to GDP. Okay. All right. And go ahead and discuss with your neighbors if you want. This clinical presentation should be familiar to you.
Oh, and I was like,
How we get biochemistry?
to the code factor.
There you go.
Okay, so everybody got this clear? So what condition, what is the clinical presentation consistent with? What's that? Yeah, glucose 6-phosphate dehydrogenase deficiency, or sometimes referred to as favism. So what's the redox cofactor for glucose 6-phosphate dehydrogenase? Okay, very good. So that's a NADP utilizing redox enzyme. Okay, great. Okay, going from the oxidative phase to the regenerative phase of the pentose phosphate pathway. I think I asked you not to remember too much about the transaldolase and transketolase, but see if you how much you remembered. Okay, I think this answer B is the best answer here. So remember the transketolase, a ketone, that's a 2-carbon molecule, right, from derived from acetyl-CoA. So it's going to take 2 carbons from one substrate and add it to another. So if you take 2 away from 5, you're left with 3, and if you add the 2 to 5, you have 7. Okay, clear as mud? Okay. Okay, very good. So this is a clinical presentation that's consistent with alkaptonuria, the deficiency of homogentisate oxidase. And the homogentisic acid elevation in the blood and in the urine would be another thing that would be associated with that in addition to the oochronosis. Good. Okay, so the PKU, the classical PKU, is caused by a deficiency in phenylalanine hydroxylase. That's the enzyme that makes phenylalanine into tyrosine. And the difference between phenylalanine and tyrosine is a OH group on the phenyl group of phenylalanine. Okay, so ring hydroxylation is the best answer here. Ooh, okay, a lot of confusion about this one. Okay, so best answer here is glutamate dehydrogenase. I think we spoke about that last week, right, in our or earlier this week on Monday. Yep. So remember, there were 3 enzymes encoded by the human genome that can fix a new carbon-nitrogen bond. We're going to talk about all of them today. So glutamine synthase, carbamoyl phosphate synthase 1, and glutamate dehydrogenase. It's a very easy test question to write for me. Okay, so. Okay. All right, let's see. So let's talk about nitrogen. Nitrogen in our body comes from primarily from eating protein and amino acids. And that nitrogen doesn't have any caloric value. We can't oxidize nitrogen and gain energy from it. So if we have excess nitrogen in our body, we have to get rid of it. And the principal way that we get rid of it is through the urea cycle. Now, a healthy adult, and for the purposes of this discussion, a healthy adult is one who is not gaining mass or losing mass. So somebody who is at a stable weight. They're going to be in a equal nitrogen balance. That is, for every atom of nitrogen that they eat in their mouth, they're going to pee out a nitrogen in urea. Okay? So there's a lot of nitrogen atoms in the body and in protein and other molecules. But when you eat nitrogen, unless you're gaining mass, or then you have to pee out all that nitrogen as urea. Okay. There are cases, and we refer to this as the nitrogen balance. So a growing child, or a woman in pregnancy, or somebody who's just increasing in mass intentionally or unintentionally, they're going to be in a positive nitrogen balance. That is, they're not urinating out as much nitrogen as they're putting in their mouth. Okay, some of the nitrogen from the amino acids in their diet is going to build new mass. Okay. In cases of starvation, essentially, all of these are cachexias, losing mass and increased heat due to, for example, dinitrophenol poisoning. Anorexia is a self-starving or not eating. And then kwashiorkor, this is a specific type of malnutrition, protein malnutrition. So not a total caloric restriction, but a deficiency particularly in protein. So in these cases, they'll have a negative nitrogen balance. Nitrogen balance is, I think the concept is important to remember. It is measured as a clinical measure, is typically used or measured in the context of somebody who is receiving parenteral nutrition, where they can't control their food, so you have to balance their diet a little bit more carefully. So it's not something you'll see very, super frequently. And to measure the nitrogen balance, it requires collecting all the urine and measuring the nitrogen in the urine, and then balancing that with the amount of nitrogen in the protein of the diet. So the formula for determining the nitrogen balance is just nitrogen intake minus the urinary urea nitrogen, and then minus an X factor. And this X factor is usually 2 grams to 4 grams, and that's going to be variable depending on the patient that you're seeing. So this X factor here is accounting for nitrogen losses aside from urea. So we do urinate out some ammonium. We also lose some nitrogen with sweat. We lose some nitrogen with gut epithelial cells that are lost in feces. So there are other minor ways that we lose nitrogen, and this X factor is kind of accounting for that. Okay. Okay. Yeah, so most of the nitrogen that we excrete every day is in the form of urea, but we also lose some, as I said, as ammonium, as creatinine, or as uric acid as well. Okay, clear? Okay, so the intracellular pool of amino acids, this is going to be in balance between a lot of other processes. So protein synthesis is going to consume the intracellular amino acids, and protein degradation is going to generate more amino acids. Those amino acids can be used or catabolized for fuel within a cell. The carbon part of those amino acids can be catabolized to fuel. The nitrogen component of the amino acid, that goes to the urea cycle, and this is a little bit simplistic in that, say this is a skeletal muscle cell, an amino acid can be catabolized for energy. The nitrogen goes to urea, but it's not within the same cell. It's going out into the blood, carried by an amino acid, generally alanine or glutamine, and going to the liver for this process. So this is somewhat of a simplistic diagram. Let's see. And I'll introduce some of the digestion and transport separately. Okay. But just to kind of get some background stuff. So ammonium is the positive ion, the NH4 plus, and that's in an equilibrium with ammonia, which is the uncharged species. And I'll probably use the terms incorrectly and just as if there's no difference between them. The basic difference, or the functional difference, is that ammonium, because of that charge, it can't cross membranes in the body, whereas ammonia can diffuse pretty freely across most of the membranes in the body. Now, because of the pK of the ratio of the ionized versus the unionized forms of ammonium and ammonia, and if you remember the Henderson-Hasselbalch equation, okay, yeah, so that's where that comes from. Anyways, so the pKa is 9.3, so at physiological pH, which is about 7.35 or something, there's going to be about 100 times more ammonium in the body than ammonia, okay, but they're in equilibrium, so they're going to, you can't partition the ammonia in one compartment and just have ammonium. It's going to be both. So the goal of most of what we're going to talk about today is really about how to get rid of excess ammonia/ammonium in the body. Ammonium are toxic. So the symptoms of ammonia toxicity are neurological. So you'll get headaches at first, that'll progress to seizures and other neurological symptoms, and it can cause death ultimately. So there's this condition or term called hepatic encephalopathy. Can you read that? Okay, so hepatic encephalopathy is referring to, it's got two words, hepatic liver and encephalopathy. That's a problem with the brain, right? So in any condition where you have liver dysfunction, because the urea cycle is one of the important functions of the liver, if your liver craps out, you can experience toxicity from ammonia, because if you don't have liver cells, you don't have the urea cycle. Okay, so hepatic encephalopathy is a general term that's referring to the neurological symptoms of ammonia toxicity due to some aspect of liver failure. Okay, now we're going to talk about some specific urea cycle disorders that will result in hyperammonemia. So that's, a very obvious cause of hepatic encephalopathy, but really you could get it with anything that's going to hurt the liver. Okay, so if the liver function is bad, either because it's got some plaque is obstructing blood flow, or because of alcoholic cirrhosis or something like that, you have the potential to develop this hepatic encephalopathy. Okay, is that everybody good on that, on the definition of that? Okay, great. Okay. So, yeah, this is the biochemist conception of the human body here. So this is, this is why they don't let me into the clinic at all. So, okay, so the digestion of protein and how we get amino acids from our diet into the blood, that's going to be a topic of a handful of lectures in the second year in the gastrointestinal medicine course. So I don't want to ruin the story for that. But just, very briefly, we eat protein, we eat nitrogen in the form of protein, and that gets broken down into individual amino acids in the gut. So the first form of digestion is denaturation of the proteins. So you lose all the quaternary structure, tertiary and secondary structure of the proteins due to the very acidic environment of the gastric compartment. Then as the protein goes into the small intestine, then it's subject to enzymatic breakdown of the peptide bonds. So it's breaking the peptide bonds to make the peptide chains shorter and shorter and shorter until they're either single amino acids or dipeptides or tripeptides. So those all, the single amino acids and the dipeptides and tripeptides, those can get taken up in gut epithelial cells. There are intracellular peptidases that will break down any di and tripeptides into single amino acids. Then the single amino acids enter into the pool of amino acids in the blood. In terms of the transport, so most of the amino acid transporters use a system similar to the SGLT system of secondary active transport for glucose. That I think we discussed that. So we have the basolateral sodium potassium exchangers that are hydrolyzing ATP and using that energy to make the concentration of sodium intracellularly very low relative to the outside. So that sodium concentration gradient then, that can drive amino acid transport through symporters that transport one amino acid with a sodium atom and can concentrate them in the cytoplasm of the gut epithelial cell. There, they can then do facilitated transport out the basolateral side and into the blood. So there are, several dozen of these transporters, and they're typically not specific for a single amino acid. They'll take kind of chemically similar amino acids altogether. Okay, so let's look at and just list to fill out this introduction. Talk about some of the key amino acids. So these are the amino acids, and then these are their deamination or transamination partners. Okay, so if glutamate loses a nitrogen, then we get alpha ketoglutarate as a product. Okay, so glutamate and alpha ketoglutarate, the basic function of those two molecules is to be the intracellular pool of amine groups and amine acceptors as well. Okay, so many of the transamination reactions intracellularly will use glutamate and alpha ketoglutarate as partners. Aspartate and its transamination partner, oxaloacetate. Oxaloacetate, I don't think that's going to come into play much in the urea cycle, but aspartate plays a key role in the urea cycle as one of the donors of nitrogen to urea. So urea has two nitrogens and then a carbon double bonded to an oxygen. Aspartate provides one of those nitrogens. Alanine alanine and glutamine, these are both important molecules for transporting excess nitrogen from peripheral tissue to the liver. Okay. And alanine, of course, is a key gluconeogenic amino acid, and there is some interplay between urea cycle activity and gluconeogenesis, as we'll see shortly. Just as a kind of a difference between these two, alanine is taking nitrogen that results from transamination reactions and giving it to the liver. Glutamine, this is taking nitrogen from ammonium, from inorganic nitrogen. Okay. Okay, so I'll try to introduce some concepts in intertissue nitrogen relationships. So when we eat dietary protein, that's broken down into individual amino acids in the liver. Those amino acids then, they can go to tissue like muscle, and insulin will promote the amino acids being incorporated into new proteins there. Okay. If we have excess amino acids, those can be used for both gluconeogenesis, and that glucose then, that can be stored as glycogen, or it can be exported out of the cell. So we know that there are some cases in the fed state where we need to turn amino acids into glucose and export them out of the liver. We can also turn excess amino acid carbon into fatty acids, and then package those fatty acids in triacylglycerol, and then into very low density lipoproteins particles for export out of the liver. And those fatty acids then can either be stored in adipose tissue, or they can be used for energy in tissues like muscle. Okay, so when I'm sorry, the liver also has a high capacity for protein synthesis. So most of the blood proteins that are circulating around in our blood are synthesized in the liver. So albumin is the major protein in the blood. But also there are a lot of clotting factors that are synthesized in the liver as well. So amino acids definitely have a function in the liver for that role. In the case where the amino acids are being deaminated, right? So there's no nitrogen in glucose and there's no nitrogen in fat. So when the amino acids are deaminated or transaminated, that nitrogen then is gotten rid of in the urea cycle, which is in the liver but not part of this um, slide. Okay, so just reiterating, I think what I said before, in the fed state, in peripheral tissue, like in a skeletal muscle, in the fed state, we're going to have abundant amino acids, abundant fat, and abundant glucose around in our body. So, depending on the energy states or the energy needs of the muscle cell, the glucose, the carbohydrates, can be stored intracellularly as glycogen, or they can be oxidized as fuel. Likewise, the fatty acids that are delivered to the skeletal muscle, either from a chylomicron or a very low density lipoprotein particle, that can be oxidized for fuel as well. In the fed state, the amino acids aren't really being oxidized for fuel in the muscle, they're being used to synthesize protein. Okay, so that's the case with the skeletal muscle in the fed state. In the fasted state, the skeletal muscle plays an important role in delivering gluconeogenic substrates to the liver. So protein in the muscle is going to be broken down, and that's going to be stimulated by fasted state hormones like cortisol. Those amino acids, the carbon from some of those amino acids is going to be used to support TCA cycle activity and be oxidized to carbon dioxide and used to meet the energy needs of the cell. So when an amino acid like isoleucine goes through this pathway, or the carbon from isoleucine goes through this pathway, the nitrogen component will be transferred to pyruvate to make alanine, or to alpha-ketoglutarate to make glutamate, and then a second amine transfer can make glutamine. So glutamine and alanine, these are the main amino acids that are transferring excess nitrogen away from muscle and to the liver. Shown here, and again, I think we're going to cover this in the gastrointestinal medicine course, but gut epithelial cells have a high rate of glutamine utilization. So glutamine, if you deaminate it once, twice, then it can be alpha-ketoglutarate and enter into the TCA cycle. So gut cells and lymphocytes of the immune system also have that type of a metabolism where they have a glutamine addiction. Some cancer cells as well have a glutamine addiction. Okay, so both alanine and glutamine can go to the liver, and they're going to be transaminated or deaminated, and the nitrogen is going to be used for urea cycle. So that's the pathway that we'll focus on. I think we've talked about ketone bodies and gluconeogenesis, so that's related because it's a fasted state process. But I'm not going to spend a lot of time talking about them today. Okay, so alanine and glutamine, these are the key transporters of nitrogen in these intertissue nitrogen relationships. But we can also get other proteins that are released from muscle, and those can also transport excess nitrogen away from the muscle and to the liver. Okay. I think that I already said this, and I think that I also already introduced this concept. So in the fasted state, you're breaking down protein in the muscle, the carbon goes to fuel oxidation, the nitrogen is transferred to alanine or to glutamine, and then those alanine and glutamine enter the circulation and bring the nitrogen to the liver for the urea cycle. So just looking at that in a little bit more detail, I'll focus first on alanine and then glutamine. Okay, so in the fasted state in the muscle, We have cortisol stimulating protein breakdown. So that's going to increase the intracellular amino acid pool. Those amino acids then will go through a transamination reaction so that they lose their nitrogen, and then the carbon can be oxidized down for making ATP. Remember that the alpha ketoglutarate and glutamate, that makes up a key amine group, donor, and acceptor pool intracellularly in muscle and in all tissues. So the alpha ketoglutarate and glutamate, that functions as kind of a nitrogen conveyor belt to transfer the nitrogen from this, generic amino acid. That the nitrogen gets transferred to alpha ketoglutarate to make glutamate, then glutamate through the action of alanine aminotransferase, that then will take pyruvate and make alanine. Okay, so it's like a conveyor belt of nitrogen going to alanine. The alanine then enters the circulation and can get taken up by the liver. There's another conveyor belt of nitrogen that's using alpha ketoglutarate and glutamate in the liver. That then is going to transfer the nitrogen to aspartate, not shown here, and then aspartate will donate the nitrogen to urea in the urea cycle. Okay. When we deaminate the alanine, we're left with pyruvate. Pyruvate then can be a substrate for gluconeogenesis in the liver. That glucose, now we know that in the fasted state, the muscle cell is not going to have a high rate of glycolysis. However, it has some need for glycolysis because it needs to regenerate the pyruvate here to keep the cycle going. Okay, is that clear? So the alanine is leaving the muscle cell, so it's got to have a way to get a new nitrogen carrier into the cell, and taking glucose through glycolysis is how it does that. Okay, so this is the generation of alanine as a nitrogen transporter, and alanine is taking nitrogen from other amino acids in a transamination reaction. Okay. That's in contrast to glutamine. Glutamine can take some nitrogen from transamination reactions, but it also has the capacity to take nitrogen from free ammonium. So we can get free ammonium in the cell through catabolism of some amino acids can be catabolized. They can lose their nitrogen by a deamination reaction that generates ammonium rather than a transamination reaction. We can also get ammonium in the cell when we're doing nucleotide catabolism. If you recall the adenosine monophosphate deaminase reaction, right? I think we discussed that as an anaplerotic reaction in the TCA cycle. So that reaction will produce free ammonium in the cell. Okay. So this free ammonium in the cell, this can be a substrate for glutamate dehydrogenase, which will take alpha ketoglutarate and add nitrogen, add inorganic nitrogen, and make a new carbon-nitrogen bond to make glutamate. So glutamate then can be a substrate for glutamine synthase. Glutamine synthase can also use free ammonium as a substrate and add it to glutamate and make glutamine. Glutamine then goes to the liver. So glutamate dehydrogenase and glutamine synthase, these are two of the three enzymes in our body that can take a free ammonium and turn it into a carbon-nitrogen bond. Okay, within the liver then, as part of the urea cycle, there are enzymes that reverse the process. So glutaminase, which is a separate enzyme from glutamine synthase, glutaminase will make free ammonium, and then the reverse reaction of the glutamate dehydrogenase reaction in the liver also generates free ammonium. The free ammonium then, this is a substrate in the liver in the urea cycle for the third enzyme that can make a new nitrogen carbon bond, carbamoyl phosphate synthase 1. And that makes urea, and that gets exported into the urine. Okay. Okay, I think we've seen this before. We're going to see it again in another context, but the glutamate dehydrogenase is not the only way that we can make glutamate from alpha-ketoglutarate. There's also the transamination routes, remember. But this is an important one. This is an interesting enzyme because it can use either NADH or NADPH as a redox cofactor. So, okay. And it's a reversible reaction. So it's occurring in this direction in the muscle to take that free ammonium and fix it into glutamate. And then it's occurring in this reaction, or in this direction in the liver, to generate the free ammonium that will be a substrate for carbamoyl phosphate synthase 1. Okay, so I think we're about ready to start the urea cycle. We've talked about how excess nitrogen can get to the liver, so now let's expand on the details of the urea cycle. I don't think I have a laser pointer, but, well, it's over there in the southeast of the blue section on our Stanford map of human metabolism. The basic function of it is, as I mentioned, to take excess nitrogen and make it into an easily excretable form. Okay. Pathologically, there are inherited defects in all of the enzymes of the urea cycle, and they all result in hyperammonemia as a consequence. Okay. So the urea cycle, here we'll get to the details of that in a moment. The urea molecule is a carbon and two nitrogens, two amine groups. So one of those nitrogens comes from free ammonium, and the other is donated by aspartate. So we can get that, we can get both of these substrates from the amino acids like alanine and glutamine and other amino acids that come into the liver through the nitrogen conveyor belt of the alpha-ketoglutarate, glutamate, amine donor acceptor system. So a generic amino acid coming into the liver transfers its nitrogen to alpha-ketoglutarate to make glutamate. Glutamate then can either generate free ammonium through the glutamate dehydrogenase reaction, or it can generate aspartate through the aspartate aminotransferase reaction. Okay, so glutamate has the capacity to make both of the substrates, both of the nitrogen substrates for the urea cycle. Okay, so I think that, yeah, I've said this, all of this, except, do you remember the TCA cycle? I think I described that as like a Ferris wheel, and oxaloacetate was the gondola of the Ferris wheel that was carrying around the two carbons from acetate. So ornithine in the urea cycle plays a somewhat analogous role to oxaloacetate in the TCA cycle. So ornithine is the start of the cycle, and when urea is clipped off of arginine, the ornithine is regenerated in the urea cycle. There's also one other intersection with TCA cycle anaplerotic reactions that I'll mention too. Okay, so this is kind of the higher level view of the urea cycle. One nitrogen from ammonium and one nitrogen from aspartate coming in. This is the figure, I think, that as you're studying, you'll want to print this one out and have it readily available for reference. This is kind of the comprehensive view of the urea cycle. So I'm just going to walk through the reactions one by one using this diagram. So we start with the mitochondrial enzyme carbamoyl phosphate synthetase 1. This enzyme is using ammonium and bicarbonate as a substrate, and it's hydrolyzing 2 ATPs to ADP. One of the phosphates that is hydrolyzed goes to inorganic phosphate. The other goes to bond with the carbon and the nitrogen to make carbamoyl phosphate. So nitrogen, carbon, and then a phosphate. And this is that third enzyme that's capable of fixing free nitrogen in our body, CPS1. The carbamoyl phosphate then, this is a substrate for ornithine transcarbamylase. Ornithine transcarbamylase takes the carbamoyl phosphate and adds it to ornithine to make citrulline. The citrulline then exits the mitochondria through a citrulline-ornithine antiporter. Okay, that's taking one citrulline out and one ornithine in. The citrulline then, this is a substrate for arginosuccinate synthase. Arginosuccinate synthase is taking the second, or the source of the second nitrogen in urea from aspartate and bonding it to citrulline to make arginosuccinate. And this requires energy of ATP hydrolysis. So this portion of the arginosuccinate is from the aspartate, and this is the part that comes from ornithine. Arginosuccinate lyase then is cleaving this bond and releasing fumarate, and then the amino acid arginine is left. So this reaction, the bonding of aspartate to the ornithine and then cleaving off the fumarate, essentially this is an amino transfer reaction, right? You're taking this nitrogen from aspartate, bonding the whole thing to citrulline to make arginosuccinate, and then cleaving off the fumarate. If you recall the discussion of anaplerosis in the TCA cycle, after AMP deaminase works to take AMP to make inosine monophosphate, there's a very similar reaction where aspartate is joined to the nucleotide to make adenylosuccinate, and then adenylosuccinate lyase cleaves off fumarate, and that regenerates the adenosine monophosphate. So just if you look back at that TCA cycle lecture and review the anaplerosis, it's kind of a similar reaction, kind of a clever way or a different way to do an amine transfer. Okay, so after a fumarate is cleaved off, then we have arginine. Arginine is a major regulator of the TCA cycle activity, and I'll touch on that towards the end of the lecture. Arginine then, that has urea right on it. So this structure here is urea. The arginase reaction, or the arginase enzyme, is making that cut and releasing urea, and when you cleave off the urea portion of arginine, you regenerate ornithine. Okay, so that's the entire cycle. And yeah, so the regulation of the urea cycle, I'll just introduce for just a moment. So essentially, it's not a super tightly regulated pathway, I'd say. So the urea cycle, the liver has a very high capacity just endemically for urea cycle activity because so many processes in our body are generating ammonia, and the consequences of ammonia toxicity are so severe that there's a lot of reserved capacity for urea cycle activity in the liver. However, the regulation of the urea cycle is largely with arginine. So arginine is, first of all, it's going to, excess arginine is going to activate and increase the activity of arginase, and that makes ornithine, and that's one of the key substrates for the urea cycle activity. There is a molecule called N-acetylglutamate that acts as an allosteric activator of CPS1. N-acetylglutamate, that's made by NAG synthase or N-acetylglutamate synthase, and N-acetylglutamate synthase is allosterically activated by arginine. So arginine acts in a couple of ways to increase the activity of the urea cycle. Not shown here, there's also glutamine acts as a transcriptional regulator of urea cycle enzymes, including arginosuccinate synthetase, and that occurs through glutamine being a nitrogen donor to make N-acetylgalactose, and N-acetylgalactose then glycosylates a transcription factor and activates the transcription. Anyway, that's maybe a little esoteric, but consider arginine as the major regulator of urea cycle activity. Okay, I think we're at our break here. Are there any questions? Okay, well, why don't we go ahead and break and come back at 2:01, and if you have questions, then I'll be happy to answer them.
The gas is in here or what?